name is Amanda Shanley, and for my summer undergraduate research program, I worked with iGEM, which is International Genetic Engineering Machining Competition, and as part of that we worked in wastewater treatment and looked at biofilms and how to optimize biofilms to optimize wastewater treatment processes. So part of that is we took two different proteins and we transformed them into the bacteria. So we have an adhesion protein, curline, which allows for formation of biofilms faster. And then after that is expressed, then we express a silica binding protein, which allows a glass covering over the biofilm. So we take a biofilter and make it a mechanical filter. So the first thing that we did was we looked at E. coli and how E. coli forms a biofilm. We grew them in 24 well plates, and then we stained them with crystal violet. So you can see here that different strains grew biofilms faster, and then different concentrations also grew it differently as well. Once we have our constructs made, we will be able to grow them again and see the difference between E. coli without our protein being expressed and E. coli with our protein being expressed. So once we've grown our bacteria in our static environment, then we can go and move into a bioreactor. One of the things that we also have done is we grew E. coli that has our different parts transformed into them on these plates, so you can see all the different colonies with our parts transformed into them. And then this is a collaborative working space, so everybody works here together. You have to be really aware that other people's experiments are going on and not accidentally mess anything up as well. And then this is our temperature controlled room. It's 37 degrees in here. And this is a bioreactor like the one that we'll be using. So it has these tubes that run all the way down, and then it has, from here, there's a feed loop that goes in to here and can feed nutrients. And the cells are all around in here, and then they adhere to this tube and form a biofilm that we can then extract from the bioreactor and test to see if our bacteria with our new proteins is actually forming a better biofilm than ones without. Part of the iGEM competition is that we want to have synthetic biology awareness out to the community. We want to standardize synthetic biology parts, and then it's also the goal is to have collaborative research environments for undergraduate researchers. So part of this team, I got to work with Dr. Rickes and Dr. Claes, who are our faculty advisors, and it was really interesting to really get to know them and work with them and see what different research they do. We also worked closely with graduate students, so we got to see what graduate school is like and some of the challenges that they face. And then we work in a group, so there's 10 people here this summer, and we're all working together on this project, and one of the challenges is organizing everybody and figuring out, you know, what exactly do we need to do, what experiments are we going to run, because there's no one saying you need to do this project and you need to do this experiment. It's all what do we need to do, what do we need to get accomplished, and organizing ourselves and setting our own guidelines to get through the summer.